Today, we're very pleased to have Ina Starskaia. She's a doctoral researcher in the laboratory of Professor Riitta Lahesmaa's group at the University of Turku. And her talk is titled Distinct Cellular Immune Responses in Children and Route to Type 1 Diabetes with Different First Appearing Autoantibodies. A little bit about Ina is she's as I mentioned, she's a doctoral researcher at the Turku Bioscience Center at the University of Turku. And she, during her MSc or Master in Science Studies, she gained in-depth knowledge in immunology. She graduated in 2012 from St. Petersburg State University in Russia. And she then went on to Turku to study the role of Th17 and iTreg in regulating the inflammatory autoimmune axis. Particularly, she was engaged in exploring the role of estrogen receptor alpha, ER alpha, in T cell differentiation in context of autoimmunity. She was also recruited to set up and optimize a new cytoph techniques using newly purchased mass cytometer at our center. Ooh, that's exciting. And she played a central role in optimization of mass cytometry workflow. She obtained experience in immunoprofiling using cytoph technology, and also is working on several research projects. She is planning to continue her academic track in the field of immunology. And at present, her research is focused on the understanding of early life immune disturbances in children who develop islet autoimmunity and identification of potential biomarkers to predict and monitor type 1 diabetes. I will share her most recent paper in the chat. I'm going to drop that in now. And thank you so much for joining us, Ina, and appreciate you walking us through your work.
Yes, thank you very much, Monica, for the nice introduction and also for the invitation to talk about our recent findings. I'm going to talk today about distinct immune responses in children who progress to type 1 diabetes with different first appearing autoantibodies. I am in the final stage of my doctoral studies, and then all my doctoral studies has been focused on early immune responses in type 1 diabetes. And I will maybe later explain you why. So, here is our group in Turku Bioscience, led by Professor Riitta Lahesmaa. And then we basically do have main direction of research. First is about novel regulators of T cell differentiation. And another big studies is focused on immune responses in type 1 diabetes. So why we decided to do these studies? First of all, as we all know, that seroconversion is the earliest indicator to predict type 1 diabetes development, but it's actually already reflects ongoing autoimmune reaction. And then it would be really nice to find earlier biomarkers. And then basically, most of the studies has been focused when they have been exploring circulating immune cells, focused on those who already developed disease. Therefore, the first question which we have, can we actually find these changes in immune cells between cases and controls very early on, so before the clinical disease, but also before seroconversion? And the next big question in type 1 diabetes is a heterogeneity. There is a lot of heterogeneity also before in the rate of progression to the disease, and also post-onset beta cell decline, but today we mostly will talk about the rate of progression before the disease. And then we know that there is a lot of variations and there are many factors which affect, but particularly age of seroconversion and type of autoantibodies and amount of autoantibodies influence a lot if the children will progress fast or slow. For example, it has been shown that if children develop multiple autoantibodies at very early, then they will progress faster if they develop multiple autoantibodies later in life. So, and then IA and GADA, usually the most commonly detected in the first appearing autoantibodies, they also differ. IA appears very early at 1-2 years of age, while GADA peaks at 4-5 years of age. And that's why we had the second question, if immunoprofiling, which we will discover, if it can be correlated with the distinct autoantibody profiles. To address this question, we have a very nice type 1 diabetes prediction and prevention study in Finland. And maybe, as you know, that Finland has the highest incidence in the world of type 1 diabetes. And in 1994, this study was established, and we screened here all newborn children. And those children who have genetic risk for type 1 diabetes, they are invited for the follow-up, and then they further monitored for appearance of autoantibody. And we also collect the samples from these children. That's why we have quite many and unique collection of the samples. So, and to address all this question, we were really lucky to have this cohort of children. We have 29 case control pairs, and all pairs have been divided into 3 groups: those who develop IA as a first autoantibody, those who develop GADA as a first autoantibody, and children who have been tested for 2 or more of autoantibody at initial seroconversion. From each child, we had 1 or 2 samples collected before seroconversion, and 1 or 2 samples collected after seroconversion, but before type 1 diagnosis. And to do the immune profiling, we decided to use mass cytometry, which allowed us to have 40 markers in the panel. And on the left, you can see our backbone panel, which has markers for discovering main PBMC subsets. And we also have added some additional markers which we thought would be interesting to see in type 1 diabetic children. So, and then we did the staining, we did the run for the samples, and then we did the analysis. So first of all, we wanted to see the composition of children PBMC, and we did clustering using lineage-specific markers. We did unsupervised clustering, then supervised clustering, and annotated the clusters based on the known biology. And we discovered 10 main PBMC subset, which you can see here on the left. And then the next question which we had, if there is a differences between the cases and controls in cell type proportion, and if these changes are specific to autoantibody group. And we applied linear mixed effect modeling. And you can see here we had age, sex, HLA, then group as a fixed effects, and batch and pair as a random effects. And then what we have found. First, if you look at the right, we can see the analysis of all samples, how we usually do without looking them in respect to autoantibodies. And then we don't see any difference between cases and controls. But then when we look at different groups of the children based on a type of autoantibody which appeared first, then we started to see the differences, in proportions. For example, in NK cells in multiple group, there is increased proportion of NK cells, in particular at the very early stage of disease development. The same we have found in GADA group. We also found increased TCR gamma delta proportion in multiple group. Next, we also observed decrease in CD4 and CD80 cells in multiple group, and decrease in P cells in GADA group. So the only cell type we can see was different and associated with the disease progression was the difference in CD8 T cells. But also, if you look closely at different groups, then we can see that actually in multiple group, there is decrease of CD8 T cells in cases compared to controls, while in GADA group, there's increase of CD8 T cells, in particular at the very early stage of disease development. So then we were kind of happy to see these results because without this classification based on the type of autoantibody which appeared first, these changes would have been masked. So the next, we wanted to see if there is a difference in functional markers expression in each identified cell type. And then we looked at them, and we also applied linear mixed effect model and looked in each of 10 cell types which we identified. And then we found increased expression of CD161 in NK cells in multiple groups. We didn't find a difference in level of expression of CD161 in IA or GADA first groups. Also, when we combine the samples, we don't see this difference. CD161 is expressed on the majority of NK cells, in particular in children, and little is known about its function, but some studies show that it is upregulated upon IL-12 exposure. And also it has been shown that CD161 positive NK cells have high production of IFN gamma compared to CD161 negative NK cells. So we further wanted to see from where the increase is coming, and then we found that the proportion of CD161 positive cells is higher in cases compared to controls. And then we also wanted to see how it looks like time point wise. And here we can see 4 time points, the first 2 before seroconversion and the last 2 after the seroconversion. And here we can see the PBMC samples and NK cells in violet color and CD161 expression. In the upper panel, this is cases, and in the lower panel, this is controls. And we can see that the signal from CD161 is higher in cases compared to controls, in particularly in the early stage of disease development. So next, we were particularly interested in CD4 and CD8 cells, and we decided to look and to look at the heterogeneity of both of these subsets. And again, we did the clustering annotations, and we have found 7 clusters of CD40 cells. Majority, of course, represented by naive cells. Then central memory cells, we also found cytotoxic CD57 expressing T cells, Tregs, and other cells, effector memory and effector phenotype. And then again, we looked at the expression of functional markers in each of these subtypes of CD40 cells. So, and then interestingly, we found increased expression of CD161 in CCR positive cells, CD40 cells, again in the multiple group. And CD161, the role of it is known better in CD40 cells at least, and it is recognized as a Th17 marker expression. So, and it was expressed higher in cases compared to controls. And which means that basically we can see that Th17 signaling is already activated early on before diagnosis and even before seroconversion. Then another interesting finding was TIGIT, which was lower, its expression was lower in cases compared to controls in cytotoxic T cells, and again in the multiple group. Further, we found increased expression of CD139 in several subsets of CD40 cells, but particularly it was quite obvious in memory Tregs and cells which has high expression of HLA-DR, ICOS, and also CD38. So perhaps this is some reactivated effector memory cells or some effector cells. And we there we could see clearly the difference between expression of CD39 in cases compared to controls in IA group. So here was the example, because I wanted to see, we were quite surprised, but that quite different expression. And then we wanted to see how it looks like in the, when we look at then this conventional way of how we look at the cells at flow cytometry. And indeed, we can see very high expression CD39 in cases compared to controls. Then when we observed all these differences, we decided that it would be really interesting to see if we can validate these findings in independent cohort of children, and we were lucky to get independent cohort, again 30 case control pairs and 10 case control pairs for each autoantibody group. But this time we had a limitation in the amount of the sample which we had, and we decided to do the validation in a targeted fashion and also using alternative method. We used flow cytometry, and we decided to do the validation for CD39 because it was quite significant finding and also quite obviously differential expression between cases and controls. And another finding which we wanted to validate was CD161 in NK cells. And there we found that we managed to validate CD161. We can see the high expression, higher proportion of CD161 positive cells in cases compared to controls. And when we look at the samples in respect to seroconversion, again, we see the difference is more prominent before seroconversion, like in the discovery cohort. But unfortunately, we were not able to validate CD39 in memory Tregs. We also had less samples in the end that we expected. Instead of 10 case controls paired, we had only 7. Here is the main results. So the first main important result, and that there is a differences in cell type proportion, and they are associated with the type of autoantibody which appeared first. And then these differences we can't see if we combine all the samples. And the second important finding of this study is CD161 elevated expression in cases compared to control in NK cells and also CD40 cells of multiple group. And it will be very interesting to follow up this further in other cohorts. So with this, I would like to thank my supervisor for the opportunity to work on this interesting and challenging project, that also all group members who have been involved, and in particularly also our collaborator, Laura Ellis Group, who did all the bioinformatic analysis, of course, deep study, our sponsors and families and clinical personnel. And thank you very much for your attention.
Thank you so much. That was really interesting. And thanks for pulling out this sort of salient points from your recent paper too. I have a question. With the, increase, one of the early slides indicated there's an increase in the TCR gamma delta. Can you comment a little bit about that?
TCR gamma delta.
Mm,
Yes, there was in multiple groups. Well, no, not really. That was quite we were not initially interested particularly in this subset.
mm.
No, I unfortunately I can't say why, but that would be interesting. We also now found from this study, because we often since we are working mostly with CD40 cells,
Mhm.
then it was maybe one of the first time, at least for me, when we look at the PBMC cells, and then we discovering interesting finding in other cell types, like, for example, NK cells and also TCR gamma delta.
And then, if looking back retrospectively on some of these data, is there a way to kind of look more granularly, kind of drill down on some of the data you already have and even separate it even more finely to understand the windows of time that these cases, over the windows of time that these cases presented, or is that not possible with this specific group?
You mean to look at the particular window, for example, before seroconversion, or like after seroconversion, but before disease? Or
Yeah. Even though I know I can see that, you've made a great case for looking at the different, the different autoantibody groups. But were you able to, within those groups, see sort of before and after?
no, we didn't see. I would say that majority of the findings which which we found, they actually belong to the very early stage of disease development. Most of them are very prominent even before conversion.
But you don't have data points sort of like along the trajectory.
Yeah, that would be maybe interesting to see. I'm not sure that we have maybe enough of the samples to look at this more clearly and to see how it develops with time, and if this is possible to find.
Okay, here's a good question from Anup Arangari. Nice talk, Ina. Based on your experience, what are the determinant antibodies or autoantibodies in children or adults that can definitively indicate T1D onset or progression? It's a question, $1 million question.
Sorry, can I look?
Sure, you can read it from the chat.
Yes. Which are the determinant antibody in children that can definitely indicate onset? Well, I mean, it's like the amount of autoantibody can indicate the progression. If we have multiple autoantibody, then the disease likely will happen, develop after some time. But then also there was a study which showed that appearance of IA2 autoantibody, especially the very early stage, will likely predict the development of disease very fast. What else? Zinc transporter 8 autoantibody usually associated with the later, it appears later. I think it's difficult to say. I think the combination of the type of autoantibody and also the age when the autoantibody appears, that might be play more important role than autoantibody itself.
Okay. Great. I wondered, well, I mean, again, he said, thanks so much for the answers. Anyone else can feel free to drop something in the chat or ask to unmute themselves.
Hey, Monica, this is
Yes.
Carla Grima. Ina, beautiful talk. Thank you so much. Jane's asking about B cells. That was my first question. The second question is, and maybe it's related to what you just answered. If you looked at the proximal visits on an individual person, do they have a very similar characteristic? In other words, does the individual kind of live in a certain space? I realize over time you also have the disease progression, but I'm just wondering if you really just looked at the ones next to each other, how reproducible in an individual is that phenotype? But also Jane's question about B cells.
I'll bring hers up next. That's a great question, though.
Yes, individuals, they usually, the phenotype is quite preserved. Of course, there is changes, especially if when they closer to disease progression and maybe after this. But otherwise, if you look at the early samples, so the phenotype is mostly the same, like largely. They preserve the phenotype.
That's very interesting. From Jane Buckner, were you able to observe differences in B cell populations as others have found increase in transitional B cells and decreased BND?
No, unfortunately, we didn't find any differences in B cells when we look at the expression of functional markers in the bulk B cells, but we didn't look into heterogeneity of B cells, because I think one of the reasons was that our panel was mostly aimed to look at CD40 cells and CD80 cells rather than B cells. But no, maybe we looked, we will try to look if we found something, but no.
In terms of the NK cells, you know, pretty obviously from this that they're playing a role. How are you planning to follow up on these data that you've kind of uncovered?
So I think, first of all, it would be nice to follow up in just other cohorts if we can see, if we can reproduce the same. And then it would be also interesting to see, to do, for example, single cell RNA sequencing and to see what are those CD161 expressing cells, what else is there. And what else? Also, I would add maybe more markers for NK cells next time, just to see, to look at the heterogeneity and then to see what are those features of those cells and how they can contribute.
What is the residence of the NK cell in the, you know, at the islet? Are they typically sort of at the PLN and then, you know, found in the islet? This is just, I guess, from pathology studies. When do they sort of show up at the islet?
Well, at least in lymph nodes, yes, and in islet, I don't know about this, but I think there was one recent study which I think they explored the lymph nodes from a patient and also autoantibody positive individuals. I think they also showed they did single cell, or maybe not single cell, but RNA sequencing, and I think they showed the difference in expression of gene for CD161, KLRB1. I think it was different, but the difference was opposite to our findings. So here we have increased in cases, but they have decreased. So maybe then.
Who was that? Do you recall who that was, that paper?
No, unfortunately, I don't remember.
That's okay. Yeah, you can There's so much to read. We'll track it down. Okay, last call for questions. Anyone else? Last call. Well, we look forward to the next set of experiments, maybe some RNA-seq studies coming from the Lahesmaa lab and from you yourself, Ina. It was great talking to you. Really interesting work, and I'm sure a lot of people will be sort of digging into this paper after they listen to this talk, and we will be editing it a little bit and then posting it up on our in our expanding library. So thanks again for joining us, and appreciate all you're doing. Have a great rest of your evening.
Yes, thank you very much.
Thank you, Ina. Bye-bye.
Bye bye.